Okay. So I think we left off here with this idea that you can have um, some special cases of microbes living within microbes in the sense that protozoa are single-celled organisms. And here you have the algae inside, lots of algae, single-celled algae. And that you can have this dynamic where typically it's a win-win. They each provide some nice uh, conditions that each one needs. And, for the paramecium, being able to go into anaerobic zones and not die is great. They're carrying their own oxygen tanks, so to speak. And then the downside is that the algae can be endangered by the idea that if the protozoa gets stressed out, some of the algae will be used as food. So I guess not ideal conditions for the algae in that condition, in that circumstance. There's some other weird things going on. So, usually we talk about how eukaryotic organisms have mitochondria. There's a few that don't. And they are primarily obligate anaerobes. So in that regard, they don't need mitochondria. If they're obligate anaerobes, that means they don't survive in oxygen. And remember, your mitochondria right now, that's where the oxygen is really working for you, because the oxygen sitting at the end of the electron transport chain in your mitochondria. And through that whole process in the mitochondria, getting you a lot of ATP to be formed. Well, if you're an obligate anaerobe poisoned by oxygen, you don't really need that going on. You don't want that going on. Aerotolerant anaerobes also can have a lack of mitochondria because they just operate through other methods. Fermentative organisms sometimes will lack. Basically, a lot of, well, a fair dose of parasites can also lack a mitochondria. They just depend on their host to provide the oxygen required and do other forms of physiology. A lot of what they are using as study organisms are showing up in rumens, primarily of cows. They usually use cow as the host. And they actually operate on the cow and actually create like a hole. I mean, that's essentially what it is. It's like a hole in their side that can go right into the rumen area of their stomach, of their intestinal tract. And then they have a cap for it and they cap it off and they keep the cow happy and healthy. And then we'll routinely uncap it and go and dig out some rumen contents. It's kind of amazing. They have that going on, I guess, at MSU. One of the former students, Dan, who took this class, I might even have pictures later on when we start looking at some interactions where he got to put his hand into the rumen himself. And so it's interesting, but at least the cow is happy and healthy, kept well fed and well cared for. So some will have this funky organelle. That's called a hydrogenosome. So hydrogen, why did they name it that way? What they see happening in this organelle is that you get this reaction of pyruvate, which is actually a substrate or intermediary substrate in glycolysis. It shows up in glycolysis. And what can happen is the reaction goes from pyruvate and it gets broken down into carbon dioxide and hydrogen gas, acetate, and I put ATP here because that's the whole goal of this reaction, is to get your ATP. So it's a fermentation reaction. And then what they see is that inside these eukaryotic organisms, there'll be methanogens living in there. So these bacteria, these archaea, that are living inside the eukaryotic, and they're floating around. And what they do is they'll take the carbon dioxide and the hydrogen gas that is getting excreted out of the hydrogenosome into the cytoplasm of the eukaryotic cell. They'll grab that and use it. And so here you have a case of a eukaryotic cell with a methanogen inside. and they're coexisting and using, you know, the methanogens using the waste essentially to get its own ATP needs met. So here's kind of a mock-up of that, where the big blue is the eukaryotic organism and the cytoplasm, and then the hydrogenosome in the brown. And so you see the glycolysis going on, and then the pyruvate gets shunted into the hydrogenosome. And then they don't really finish the thought here, but here you have the methanogens then taking that and making methane off of it. So some interesting examples of organisms living inside and like directly in the cytoplasm, which is pretty unusual. That usually means there's been a long coevolutionary history amongst these two organisms to allow for this dynamic to be persisting and coming into a stable mutualism. Because usually if something gets into your cytoplasm, you'll want to try to kill it. And so there had to have been a long dynamic to kind of get this situated out where they are capable of coexisting. So cooperation is a fun idea to think about, and actually has met with some reluctance on some scientists to use this term when you think about some of the microbial ecology interactions going on, but probably it has gained the most acceptance in the example of the slime molds. And so you have the cellular slime molds that are eukaryotic organisms, microscopic, until they come together. And so the classic example is Dictyostelium discoideum, that slime mold. That's kind of like the guinea pig of slime molds that's been studied. And what they do is, under typically stressful conditions, the individual cells that are just kind of doing their thing will start to aggregate together. So in order to do that, they got to find each other. There's quite a bit of communication going on between the individual cells so that they can find each other and start to aggregate or come together. And then they start to migrate as a mass unit, looking for food and kind of hunting together as a team. If they start to run out of food, they'll switch into doing a fruiting body formation. And this is multicellular, so it's a joint cooperative effort to get this fruiting body architecture made. And what you'll see is there'll be some of the cells will be attaching to the substrate of whatever they're moving across. Maybe it's a rock or a dead tree trunk or something. And then you'll start to see typically some of the cells forming some kind of stalk structure. And then at the top of the stalk, you typically see that's where they put some cells that will develop into spores. And so here the idea is, okay, we've come together under tough times. We're trying to hunt, can't find anything. Really, we're starting to starve. So let's switch into fruiting body so that at least some of us survive. Let's get some of us going into spores where we can go dormant. And so this idea of cooperation where it's a group effort in order to get this to happen. So this has been really heavily studied because there's a lot going on here with regard to the ecology of these organisms. And here's a mock-up of the different stages. They've named the different stages as well. And so if you start from, you know, an individual cell, like the spores germinating down in the lower right here. And then you can see that they start to grow out from that. And then they start to aggregate under tough times and start to form the fruiting body. So sporocarp is just, or saurocarp, I should say. I say fruiting body. Most other places say that as well. So this has been well studied. Here's another image trying to show the timeline. So you can see, you know, in this case, they were showing about 8 hours under experimental conditions. And so they think the stalk is formed so that the spores are up high off the substrate, relatively speaking. You know, these are very tiny organisms, so but get the spores up off the substrate so that with wind dispersal or something like that, you'll hopefully have the spores getting into a habitat that is not devoid of nutrients or water. Here's an actual timeline with real pictures. Migrating slug. And it really does look like a slug. The Mexican hat stage. I like some of the names of the different stages. And then by the time it's all done, you have that nice tall stalk with, so it's in the sorus region. Anytime you start hearing sorus, you start to think about there's must be spores inside there. That's a typical term. We use that with ferns as well. When you look at the underside of fern leaves, they have sori, where there's a bunch of fern spores. So the spores are nice because they're relatively resistant from harsh conditions. And again, they can go, you know, they're dormant, so they can wait out this tough time that all the other cells were experiencing. And so it's a good strategy. Some of those cells, I mean, you know, most of the fruiting body, if those tough times continue, they're going to die. So they've also thought about the idea of altruism, that some of the cells are kind of sacrificing themselves for the good of the entire, you know, individuals there. So there's been thoughts about that as well. That's not talked about as much, but it's an interesting idea to think about. Here's another really nice image showing the process. And this one, this was an interesting study because they were finding when they started to look at the slime molds in the sorus region where the spores are forming, bacteria are getting packed in to the spores. So these are eukaryotic organisms, okay? They're slime molds, they're not quite fungi, but they're eukaryotic. And this study was showing that they actually pack bacteria in with them so that when those spores germinate and grow, they have a food source right away. And so it's an interesting idea that it's a way for them to kind of prepare for when, you know, because when those spores germinate, maybe it could be maybe a very brief time of good nutrients, but then it might quickly go downhill. And if they pack some bacteria with them, at least they'll have a little bit to tide them over, hopefully, and make it through. So lots of active research going on with the slime molds. A very, very interesting group of organisms. Here's a beautiful picture that entered into a contest where you can see the sori there, where you can, if you were to crack into that and look, there'd be a bunch of spores. Here's another really cool one. Beautiful images. These, I think, were entered into there's a small world competition where photographers take pictures of really small things and enter it in for competition. So
yeah, is this using microscopy or just like a micro lens?
this one, it says the technique was stacking. The image was stacked, which I don't quite, and they said they use reflected light and the magnification is 10x. So that's not much of a magnification, really. That's what they did for that one. I don't know what, with this one, this is a 10x as well, 10x magnification. So
Acting as a technique of shooting the same composition multiple times with various focal lengths.
oh, cool. Yeah, I'm not a photographer, so I can certainly admire their resulting pictures, but I half the time I'm like, how do they do that? So here not quite as pretty, perhaps, are myxobacteria. So the slime molds are eukaryotic, but there's also a group of bacteria called myxobacteria that kind of do a similar thing, except of course they're going to be much smaller because they're bacteria. But basically, they come together and will cooperate. Again, they mass together. So this A, B, C, D, E is trying to show the progression of, you know, A, they're more like on their own, hanging out, and then they start to come together. Figures C and D in particular, and E, you can see how they're massing together. And again, the same idea, they'll come together, they have to find each other, so they're communicating with each other, being able to find each other, start moving as a unit, looking for food to break down. If everyone is chipping in and excreting their enzymes, it's easier to digest really complex substrates. So you tend to find myxobacteria hanging out on that kind of thing, like tree trunks where you have a lot of lignin and hemicellulose and cellulose. And so that's a good place to hunt for myxobacteria because they're capable of working as a team to more efficiently break down that kind of complicated and resistant molecules that take quite an effort, lots of different enzymes to break them down. So again, the fruiting body, not quite as dramatic, way smaller. You'd need more than a 10x microscope to see them. But again, they're trying to show this progression of, well, if you start again from the spores, germinating out, you know, if times become good, they'll germinate out and start growing. Times go bad, then they start to aggregate and come together. And so again, if the bad times persist, chunks of the fruiting body will die off and only the spores will make it into the next generation, so to speak, or through the tough time, I should say, and be able to persist. Okay, so those are some interesting interactions that can go on that have been heavily studied. Is the next slide set in EduCat? Is there one called anything else showing? Okay, sorry about that. Thought I had it in there. Let me post that in there. I updated it and I apparently forgot to update the EduCat. Okay, there should be one. Did it show up? Where'd it go? Oh, there. Okay, if you refresh EduCat, there should be one called Species Interactions -QS. So that's the next one I wanted to continue on with species interactions, but this is kind of a more of a look into how we get these interactions to actually occur in terms of in order for interactions to occur, sometimes what has to happen is you have to have individuals recognizing that there's another individual nearby, and then they have to be able to communicate in some manner. So this has been a topic that's kind of late in coming, in the sense that it's a pretty new idea. It hasn't been around that long, primarily because, well, first of all, a lot of people thought there's no way microbes could be this sophisticated and actually communicate in such a manner. So there was that kind of just resistance to the idea. And then the other thing was just the technology wasn't quite there yet in order to demonstrate that this is occurring. So quorum sensing is something that I looked at when I was a postdoc, working at the University of Minnesota, because we were trying to design biocontrol agents that could inhibit or kill a certain plant pathogen. And we were interested in using mixtures of biocontrol agents that cooperate and work as a team. And with that idea, it kind of makes sense that perhaps some of them are actually communicating with each other. We had evidence to show that. And so we started looking for this idea of, can they chemically communicate with each other in order to move into a behavior that suits both or all of the partners that are involved. So quorum sensing, I'll talk about that in some a little bit. And then we'll get into these other ideas that are even newer than that in terms of just the research is pretty new on nanotubes and extracellular vesicles. And so if you have all these microbes hanging out, and now the general consensus is that in natural habitats, microbes hang out in groups. And if that's the case, kind of makes you wonder, are they talking to each other in some way? Are they able to figure out who's around them? Are they able to, come to, and there's plenty of ideas. We just talked about how we see that they can team up and work as a team. They can cooperate. So how are they doing that? They must have some way to communicate. And so they started searching for this. They used, this idea, of course, they don't have vocal cords and talk audibly. It's through chemical communication. Although there have been some scientists who say that some microbes do use sound, but that's a different topic. That's very, very ephemeral yet. There's not a lot of data on it. But anyways, this definitely has some data. The idea that there's chemicals that microbes make, and then those usually get sent out into the surrounding, and it's used as a signal to other microbes nearby. And they call it quorum sensing because if you think about, if you've ever belonged to a club or some association, you have to have a quorum in order to vote. So you have to have enough people present in order to, pass a vote, for example. So this idea of a quorum, what they're trying to say is that generally you get this quorum sensing going on when there's a lot of individuals hanging out together. And so you have a certain level of population of these organisms, you have a quorum. They've also been called pheromones. That's taken more from the animal and human research idea, talking about pheromones amongst animals. So sometimes you see that word. And then another word that they use for quorum sensing is autoinducers. So all of these you'll see in the literature. But quorum sensing, I think, is a fine way to think about it. I'm not going to go into huge detail. I do talk about this in Gen Micro a little bit more detailed in terms of the physiology of it, but I'll mention a few things just to get you familiar with how it works. But basically, they started looking at, because gram-positive and gram-negative bacteria, their cell types are very different from each other. They wondered if they have different ways of communicating. And in general, the answer is yes, although it's not quite so cut and dried. So we tend to, when we teach this, we tend to talk about how do gram-positives do it, how do gram-negatives do it. But since more research has been done, we realize that it's kind of a gray area in the sense that gram-positives have multiple ways of doing it, and so do gram-negatives. So it's a little murky here, but let's go with the idea that there's some defined differences between them, and there are, and I'll talk about that a little bit. And the reason I have this little cute aquatic squid is that this whole idea of quorum sensing was first discovered in Alivibrio, which used to be called Vibrio fischeri, which is a aquatic bacteria. And it hangs out with squid and a bunch of different fish. And if you played it out onto an auger plate, and you just look at it under normal, fluorescent lights, it would just look like what you see in figure C. But then if I told you to walk into the warm room in the lab and turn off the light in there and let your eyes adjust, you would see it bioluminescent. And the colonies would show up just to the naked eye, they'd be glowing. And so Vibrio fischeri is famous for that ability. And you don't need any special like a black light or anything like that. It just shows with the naked eye. If you're in a really dark area, you can see it. So they're really cool to work with. And they've been renamed to Ali Vibrio, but they're gram negatives. And they were first isolated from the bobtail squid because the squid was bioluminescing and they were trying to figure out what's that about. And so they are the first organism to know that they studied where they learned that this quorum sensing was going on. So in the big picture idea, what we have is this mechanism where the bacteria, so Vibrio or Ali Vibrio, is busy making a chemical, a quorum sensing chemical. And then it excretes it out of its cell. So that's what you're seeing here in the synthesis idea. And that it's made and being excreted out. And then what they're trying to show is that a nearby cell recognizes that. And the simple way to think about it with gram negatives is that those signals, you can see this purple thing and this green thing, those are receptors, protein receptors that are in the cell membrane of the bacteria. And you can see these chemicals are docking into those receptors. That's what they're trying to show here, that they're making contact with the receptor. So these quorum sensing chemicals are interacting with those receptors. And then they're trying to show that that is the signal that can bring that information into the cell. And ultimately, once that signal is transduced into the cell, it ultimately goes to the DNA and gets the DNA to turn on or turn off certain genes. Okay. So then what? Then there's some kind of response. And it really depends what habitat you're thinking of, what microbes you're thinking of. The response can be a lot of different things. But in this case, they're showing maybe virulence, maybe bioluminescence, or other group behaviors. And I want you to key in on that, group behaviors, all right? So basically, what quorum sensing allows for is that when these microbes come together, they can do something as a group or a team much better than they could do individually, or maybe they just can't do individually. Probably the best example I can give you that unfortunately we're all aware of is when you get sick. So let's think about strep throat. Strep throat is caused by Streptococcus pyogenes, nasty little bacteria that loves to attach to your throat cells. And initially, when strep gets to your throat area and starts to set up camp there, you don't know you're sick yet. But they're very busy. They're busy doing this idea. They're sending out chemicals. How many other strep pyogenes are around me? Is there anyone else here? Oh, no one here yet? Okay, I'm just gonna kind of try to hide away. I'm gonna try to get some nutrients. I'm gonna try to reproduce. And they keep assessing how many of me are around here. How many? Because if you think about it, you're such a sophisticated organism. You have a really good immune system. If that strep pyogenes, if there's only like 10 of them, they're not going to try to attack you, right? That would be not wise because your immune system would zap them in a second. Let's do it. So they lay low and won't do any virulence behaviors yet. But then once there's 1,000 of them, and they're all, constantly sending out these chemicals, and through this buildup of the chemicals, they start to see, oh, there's a lot more around here. There's a lot more. You've reached, they often say, the threshold. the threshold has been reached in terms of the population size. Now it makes sense for them to enter into a group behavior because they have a better chance of beating your immune system if there's a lot of the cells. So think about warfare. If you have a large army, better chance of defeating the enemy. So this idea of wait, keep assessing, figure out how many strep pyogenes are around. Don't do any group behavior until you have a chance of success. That makes sense? Okay, so this idea of group behaviors. And, you know, what's really happening is at the DNA level, eventually when they say, okay, let's go, then the certain genes for like virulence get turned on, and then they start pumping out the virulence factors, start attacking you. Then you start to feel really bad. You get that terrible sore throat, you pop a fever, and it's all on war in your throat area. Okay, so these chemicals, again, they're quorum sensing chemicals. They're also called autoinducers, pheromones. If you really want to know at the base of it, it's this. This is the chemical. So they're the group, I should say. It's a group term. So they're N-acylhomoserine lactones. And often in the literature, they'll abbreviate it down to HSL or AHL. So these are the quorum sensing chemicals. And so what's going on is that the bacteria is making that inside, and then they excrete it out. And then as a bunch of these, like the strep pyogenes, if there's a bunch of them all together and they're all doing this, you're going to get that signal, higher and higher doses of the signal. You get this higher concentration. Yes, yes, yeah, so they're all like hanging out in that area and they're all sending them out. And then if you get a lot of that signal, then they're all, you know, starting to interact with the cells around. And that's how they go, oh, there's a lot of signal coming at me. I know that there's a lot of other cells around. And then that's when they kick into the group behavior. So in order to do that, they'll shift their gene expression to turn on virulence to start attacking your tissues, for example. So, did you have a question?
Signals here looks like self-interact, and how do they know that those signals are from themselves versus?
Yes, so the autoinducer term that is given, that's actually getting at that idea that there will be, you know, because it's self-made signal, right? And then it's coming back at them. But the idea here is that when they produce a signal, it's a very low dose. Just a very low dose, very low dose. So if they get this high dose coming at them, it's this dose dependent thing. And so if they, that's why they often use the word threshold. The threshold concentration has been exceeded. That's what they're getting at, is that you have to have a lot of it coming at them for them to go, oh, okay, now we can alter gene expression. So that's what the chemical actually looks like. The lactone is the lactone ring, and then there's always a nitrogen here, and then the side chain, you put an R on the end. That means that the side chain can vary in length. There's different things that can get added on. So for example, here are some homoserine lactones, and the R chain could be just like a methyl group, or you can have a bunch of additional carbons added on. Okay, so that side chain can vary. It depends on who's producing them. And so you can see different bacteria have slightly different homoserine lactones, which are the quorum sensing signals. So we'll come back to that idea. Here's another one just showing, again, the variety. It's the same basic idea. It's a homoserine lactone. It's just that that side chain can vary. And so that's raised some interesting questions about, you know, can these, for example, can Pseudomonas understand what maybe Vibrio is making or Rhizobium is making? So this idea of, can they understand similar chemicals, but not quite the same? And we'll talk about that as well. So in the case of bioluminescence, when I say altered gene expression, this is what I'm trying to get you to think about, where at really low cell density, they're just kind of, a little bit of that signal is being put out. But if you imagine 1,000 of these cells all doing this, then you're going to get a buildup of that signal in the environment, and it's really gonna come at the cells. They're gonna realize, oh, okay, we have a lot of cells doing this. We could enter into a group behavior, and that might be a really good thing. And in this case, it's for light production. So now that the gray arrow down here is thicker, they're trying to show that that section of the DNA got turned on. And in this case, it's allowing for bioluminescence, which is really cool. So bioluminescence on Petri plates. And again, just with the naked eye. I had a student in this class, Steve, when he had to do his microbial ecology research, he called his buddy out west that's an avid fisherman and had him overnight. I can't remember what the fish was. It was a commercial fish, that you would eat. I can't remember the species. Anyways, what he did was he shipped it over on dry ice. And then Steve did a bunch of swabbing of the fish, and he was able to isolate a bunch of the Vibrio off of it. It was really cool. And then he did some different studies with it. So, and again, you didn't need any special equipment. You just needed a dark room in order to prove that they were bioluminescing. So the bioluminescence is like the classical idea of this idea of quorum sensing. I just wanted to mention with gram-positive bacteria, they have a slightly different method of doing this and a different signal chemical. It's a peptide instead of a homoserine lactone. So gram-positives, what you're seeing in these figures, those are just a bunch of amino acids strung together into short little chains. So they're peptides. And so they call them peptide pheromones. That's the catch term for them. But it's the same basic idea. These bacteria are making these little peptides and they're excreting them out. And they just constantly do that at a very low, amount. And then if there's a bunch of all these cells doing that, you'll get that threshold level. And then they'll start to interact with the cells. The chemicals will interact with the cells. And ultimately, again, that signal gets brought to the DNA and will alter the gene expression to let them do some kind of group behavior that's important. And so this is something that when they look at, okay, under what conditions are they doing this and shifting their behavior? Well, they point out it's probably a way for them to figure out, who else of my species is around me. It's a way to determine your own population. It's a way to switch into a group behavior that's a win-win for everybody, hopefully. And so really, when you get to it, it is a way for them to cooperate with each other. And to be able to, in order to cooperate, you got to figure out who's around, and you got to be able to talk to each other in order to cooperate. And so if you think about some of the cooperative events, here are some that are being listed out. So you can see that there's a lot of different bacteria here. So since this was first discovered, initially they thought maybe it's just Vibrio, because bioluminescence is pretty, amazing, and not many organisms do it. So they thought it might be unique to bioluminescence ideas, but they realized fairly quickly, oh no, all kinds of bacteria are doing this. So it could be for biofilm production, virulence, plasmids, to get plasmids sent over from a donor to receptor, swarming motility, antibiotic production, aggregation of cells to come together. I mean, there's all kinds of sliding motility, production of exoenzymes. So there's all kinds of behavior that has been pointed to the saying, okay, in order to do this, they are doing this quorum sensing process. And so this is something that's gained a lot of momentum because this has a lot of applications for us and consequences for us, both good and bad. So All right, we'll stop there and I'll talk a little bit more about this and nanotubes and vesicles tomorrow.